Hello, this lecture will cover transcription and translation as it relates to our DNA to RNA to protein. So let's talk from the beginning. First, we want to ask ourselves, how do genes determine traits? So we know genes are going to be the instructions that our body gives us to give us the eventual characteristics that we have. So genes are the genetic material. In essence, this is your DNA. And it's going to be the blueprint for an organism's characteristics. So if you have brown eyes, if you are 6'4", if you have freckles, all these things are going to be based off some instructions or genetic material that you have that tell you what you're going to be. So these genes, this DNA, genetic material, will be the backbone for building a polypeptide. So if you remember, that's the polymer for a protein, a polypeptide. Those polypeptides will form primary, secondary, tertiary, quaternary structures to then give you a functional protein that's going to have some type of role or job in the cell. We remember the shape of that protein structure is related to its function. Think of a wheel that has the ability to roll. So the protein is going to have a particular shape that determines its function and how it works in the cell. And the protein will eventually give us some type of trait or characteristic, right? So once we have all of our proteins together that are working in your cell, they're going to give you a trait or characteristic that is meaningful to do, whatever your cell is designed to do. So that's how we're starting, right? You have your DNA, which is going to make your polypeptide. That polypeptide will fold to create a certain structure, which has a function in your cell, and then that's going to give you the freckles, the brown eyes, the 6'4" height. So genes can be expressed in two types of ways. One is molecular function. So that's going to be exactly how it operates on the protein level. So, molecule, let's say it's supposed to produce sodium, potassium, right? This is going to be what it does. And then finally, it can help to create a trait, a phenotype, which we'll talk about later. So maybe you have brown hair, maybe you have long nails. All these things are going to be a trait or some type of phenotype. But there are also two different ways that genes can be expressed. One, which would be protein coding genes. So these are going to be the genes in your DNA that actually make polypeptides. So they're going to make the long string of primary structure needed to make a protein. And then you have some other types of genes that are called non-coding RNAs. So these do not make any polypeptides. This is going to be very, very critical to just kind of protecting your DNA. They don't really make a protein itself, but they're still going to be critical in helping with the structure of your DNA itself. So, central dogma of biology. We talked about this in one of the other modules before, but I'm going to revisit this idea. So, DNA leads to RNA, which leads to protein. So we've already done the DNA replication piece. We've already replicated our DNA, right? This is our DNA. We've already done the replication process. That's where we had leading and lagging strand. But as we go from DNA to RNA, this is going to be what we call transcription. So the way I like to think of this is I like to think of your DNA as a big old recipe book. I'm probably spelling recipe wrong. Sorry. Forgive me. So think of a big old recipe book. It has 1,000 recipes in it. Imagine you wanted to make sweet potato pie. Right? If you wanted to make sweet potato pie, you would take out the recipe card that is only for sweet potato pie. Only. Sweet potato pie recipe. That is what going from DNA to RNA means. You have a lot of information in this big recipe book, but you want to take out just the message or just the RNA that you need to make that one recipe. That's what transcription is. Transcription means writing. It literally means writing. So as we produce an RNA copy of the gene we want, we call that an RNA transcript. That's going to be like writing down that sweet potato pie recipe. Wonderful. So now we have our sweet potato pie recipe. From there, we want to actually make the pie, right? The recipe is going to be words. You know how to write. And the pie is going to be a product, right? It's going to be an actual item that we made. A pie is much different than words. That's exactly what's happening in translation. So translation, or as we go from RNA to the protein, that is called translation. That's where we make our actual polypeptide. That's where we make our product. So the same way we were able to take our our recipe and then make a pie, that's what we're doing here down in translation. If you know what it means to translate something, let's say you know how to speak fluent French and you can translate that to Spanish. That's going to be taking something in one format, changing it to another format. So this is going to be the process of actually making the cake, making the pie. So make sure you're very familiar with DNA to RNA to protein, the terms that we're using here, what replication is, transcription, translation, how they all play a role together. That's really critical. Following that central dogma of biology, let's look first here in eukaryotes, which is going to be here, our more complex cell, and then also in our prokaryotes, which are going to be something like bacteria. So eukaryotes have an extra step called RNA processing, in which we can kind of modify that RNA a little bit so that it can be active. So I'm going to just walk you through what's happening to remind you. Your DNA is going to be housed in your nucleus. Here, you're going to undergo transcription, so you're going from DNA to RNA. You do have some RNA processing that still happens in the nucleus. And then, through that nuclear pore that we talked about before, that mRNA is going to come out into the cytosol, in which it comes across a ribosome and it is translated. So you remember how we always talked about ribosomes make proteins? This is because it can take the message, make proteins using that message, okay? In prokaryotes, we don't have as much of a complex system because remember, we don't have any organelles. So your DNA is present, your DNA makes your RNA works with your ribosomes, you get a protein. So it's very, very simple in bacteria. So again, this is just reinforcing that idea. The cytosol, everything happens there in the bacteria, and then for eukaryotes, it's the nucleus first, then the cytosol. Okay, so let's talk about transcription for a bit. Transcription is where we make that RNA copy of a gene. In that RNA copy, we call it an RNA transcript. So again, DNA, transcription, RNA, translation, protein. There are some viruses that can do reverse transcriptase, so that means they have RNA and they can go back to DNA, but our normal flow is from left to right in this case. So we can make a lot of different products from our RNA. And some of those RNAs have very specific names, which we'll talk about each one. But the one that's most important for carrying that information is called mRNA. That M stands for messenger RNA. So with the transcription RNAs, let's talk about a few of them and what their goals are. Again, mRNA is the messenger RNA. This is going to be the one that actually has the recipe down or the message for making the protein. So that's the one that says, has the code that we're trying to transcribe, that type of thing. tRNA, the T is used, actually it stands for transfer. So the tRNA is going to help to carry the amino acids needed to translate that mRNA into polypeptides. So what we're going to eventually do, which I'll show you in a second, is each of these little pairs, we're going to pair them up with a specific amino acid, right? So we're going to pair those three up with a specific amino acid, those three up, and then eventually we'll link them together to form our string of beads that we've talked about in our protein days. I hope y'all remember the necklace that I would talk about or use. So the RNA that is going to transfer or carry that amino acid to the mRNA or the message at the bottom, that one is called tRNA, transfer RNA. And then finally, rRNA, the R stands for ribosome, and this is going to form the ribosome. So the ribosome is essentially made up of rRNA, and this is going to be where that transfer occurs. So you see the big blue ribosome in this image. This is going to be where we're bringing together our code or our mRNA with our transfer RNAs. They're going to form a pairing, which we'll talk about in a little bit. You see their complementary pairing. So A goes with U, U goes with A, G goes with C. And all of these are going to pair together or bring together amino acids so that we can eventually form a long chain or a polypeptide of our amino acids. So as we go from DNA to RNA, right, let's talk a little bit more about that process. There's a few components on our DNA that are going to be critical as to how we start this process. The first one is a promoter. So a promoter is going to be where on the DNA we start our transcription process. So we're trying to go from DNA to RNA. A promoter tells us where to start. Think about a party or an event you've been to on campus. You've heard of maybe a party promoter. They're going to say, hey, Friday night, this is where you want to be. So a promoter on DNA is going to do the same thing. It's going to show you where we are starting. A terminator is the end. Seen the movie The Terminator? You've heard of the word terminate or termination from a job or something. This is going to be where we're stopping our code or stopping the transcription of our code. And then finally, you have some regulatory sequences. They're going to be towards the beginning before the promoter. This is going to help us control whether we're making this mRNA or not. So let's say you don't want to make this transcript anymore. You might have a big old protein that sits right here that blocks the transcription from occurring. That's a regulatory sequence, something that can sit here to block the party from starting. Think of like, I don't know, the fire department shutting a party down that the promoter said it's going to be at. I don't know, whatever you want to think of. And then finally, the enzyme that's going to make the new strands of RNA is called an RNA polymerase. Don't confuse that with a DNA polymerase, which replicates our DNA, but this is an RNA polymerase. So let's talk about this transcription process as it relates to bacteria, okay? So as it relates to bacteria, let's talk about it. It's going to be a lot easier with bacteria. We got 3 different steps we're going to cover throughout the process. Okay, so the first step is called the initiation phase, the very first one. This is where we recognize where we're going to start this entire process. So this is going to include a special protein called a sigma factor, which will first bind to the RNA polymerase and to the promoter sequence. So the sigma factor is right here. It's going to be kind of the bridge between the promoter region on the DNA and the actual RNA polymerase. Think of like a primer for DNA polymerase and how that's necessary for the DNA polymerase to get on the strand. Think of your sigma factor as what is required to help the RNA polymerase get on the promoter. Okay, so this is going to be a factor that's needed there. And again, the sigma factor is going to help that RNA polymerase bind to that DNA sequence, right? We're taking our DNA and we're trying to make RNA from it. This is going to create something called an open complex. So this open complex kind of opens up the DNA strand so that we can make our RNA sequence from it. So then the next process is elongation, okay? So this is just as simple as it sounds. We're trying to basically make our mRNA transcript from our DNA. So RNA polymerase is going to synthesize the RNA transcript. During this process, the sigma factor is released and your RNA polymerase will just slide along the DNA strand. As it's sliding along the DNA strand, it's using that DNA strand as a template. So it's going to be the template that it needs to be able to make the RNA. So I'm going to give you an example. Let's say our DNA strand is, let's say ATTGCC, right? That's the template strand. The opposite end of that DNA, let's say, just to give you some practice, 5' to 3'. The opposite end of that DNA would be 3 down here, 5 down here, and our numbers would be something like TAACGG, right? So let's say we're using that top DNA strand. That is going to be called our template strand. The opposite DNA strand is called the coding strand. So this one here would be the coding strand. So remember, DNA has 2 strands, it's double-stranded. So the top one, the one that we're trying to actually copy, which is the top one, this is going to be called our template strand. And this DNA strand that goes along with that one is called our coding strand. This is important because as we're making our new RNA, right, we're not replicating DNA in this process. We're trying to make RNA. That RNA strand is going to have essentially the same code as the coding strand, but instead of T's, we're going to have U's, right? So it would look something like this. So if you see, those are going to be the same strand except for the U and the T switch there, okay? So I just want y'all to see that. But even if you don't have the bottom strand, or even if you have a problem like this where you just get the coding strand, you can either solve it by writing down the opposite DNA strand and then just making that simple switch here between the T's and the U's, or you can just do your normal pairing between the template strand and your new RNA strand, making sure that you do not put any T's, but instead you put U's. So that's just a way to solve that problem if you ever get it and you want to look through something like that. Okay, so the final part of transcription here is after we've done our initiation phase, our elongation phase is termination. Real simple, right? So once the RNA polymerase reaches the terminator, or the very end, right? This is the very end of the the DNA strand. The new RNA that we made is going to separate or dissociate from the DNA, and we're left with this beautiful completed mRNA transcript. So the process is not too bad. Let me go back and just show you from the beginning everything we went through. We had our initiation phase in which we had our sigma factor that helps us bind to the promoter and the RNA polymerase. Then the RNA polymerase will move down the DNA strand to code. This is just kind of showing you some of those terms we talked about: template strand, coding strand, all that. And then finally, once it gets to the terminator, it separates and you have your completed mRNA transcript. Okay, so eukaryotic transcription, I'm not going to ask a lot of questions on it, but I will briefly just mention it. It has very similar features to prokaryotes, except we have a lot more steps. So one big thing here is that we have multiple types of RNA polymerase enzyme. So for instance, RNA polymerase 2 is going to actually transcribe the mRNA. In RNA polymerase 1 and 3, this is going to make those non-structural genes that I talked about earlier. Mostly genes that are needed to make other molecules of rRNA or tRNA. We have to make them from somewhere. They also need a code. But RNA polymerase 1 and 3 will be responsible for that, while RNA polymerase 2 is going to be what actually makes our message RNA, so our mRNA. Also, we're going to use a lot of transcription factors that help us say when we should start, when we should stop. Again, to reinforce that idea with RNA processing, once you have bacterial mRNA, we can immediately translate it and make the protein, but it doesn't always happen in eukaryotes. So I'm going to talk about a few of the little things that happen in eukaryotes that make it different so that you're aware, because remember, we're more complex. So our mRNA is usually a lot longer, and that longer mRNA is going to first be called PreMrna. So once we have our DNA up here, the first thing we transcribe is an RNA called PreMrna. That's the first one here. We're going to eventually cut and dice this thing up to make it functional, but the first level is called PreMrna. So in PreMrna, you have two different types of phases, or two different types of areas. One is called an intron. So this is something that we transcribed or wrote down, but we're not going to make a protein from it. Then you have something called an exon, which is something you transcribed, you wrote down, but we're actually going to keep it. So in this picture, the introns are green. So this is going to be the intron. And then the exons are going to be yellow. So there are more complicated reasons as to why we would write something down and not use it. But as of right now, I just wanted you to understand that there's some things we write down that we don't keep, some things we write down that we do keep. So the process of removing those introns is called splicing. So So I don't know if you've heard of the term splice, but splice means to cut or chop or whatever. So this is going to be the removal of those introns. Throughout the splicing process, we're going to go from something that has introns and extrons up here. So it's going to be yellow and green. We're going to cut it up. And then we're going to eventually end up with our mature mRNA molecule that we're ready to have, that will be able to make the protein that we want. There are a few other modifications that we have called tails and caps, which I'm going to mention in just a second. So I want to show you here, there's a 5' cap and a 3' tail. So what is a cap? A cap is just this modified guanosine that's attached to the 5' end. So to help protect your mRNA, think of like a bumper on your car. Your car has a front bumper and a back bumper. The front bumper is going to be our 5' cap. And it's going to be kind of this little methyl group. It's right here, really complicated. I'm not going to have you identify it or anything. But it's going to be on the front part of your mRNA. And this is going to be helpful for allowing that mRNA to actually leave the nucleus and go to the ribosome. And then on the 3' end of that mRNA, you have a poly A tail. So this is going to be about 100 to 200 adenine nucleotides that are added to the 3' end of your mRNA molecule. And this is going to be helpful because it increases the lifespan and stability of your mRNA once it goes in the cytosol. You don't want it to leave the nucleus, go into the cytosol, and then be degraded. And then now you're missing some of the message that you had. So this is a buffer. In case they start eating away at mRNA, it will start attacking it from this side, and you'll still have a lot of space before you actually get to your code. Again, this is splicing. Don't worry about the details associated with splicing. I'm not going to ask any questions with this. So in your notes, if you have them still in your notes, you can definitely just cancel this out. This is another concept about splicing. It's called alternative splicing, which just means if we have 4 different parts, so red, yellow, green, and blue, depending on how we cut them up, we can make different proteins. So if we just kept the red, yellow, and blue parts, you can make this red, yellow, and blue protein. If we kept the red, green, and blue parts, we can make a red, green, and blue protein. So it's just talking about different ways you slice and dice the mRNA. You can lead to different proteins. Okay, so this concludes the transcription portion of the lecture. So I'm going to pause a little bit, and I'll probably stop the recording now just to give you 2 separate items to review.